Hello everyone, this is Shauna. I'm presenting on Laurussus tenuis, the California Grunion, and its reproductive and early life adaptations. And I'm sorry for the loud background noise, but I am on an airplane. So the Grunion are best known for their spawning event. Their genus Laurussus is the only genus in marine teleos that spawns on land. So between March and August of every year, the adults come up onto the beaches a few days after the new moon and full moon, riding the high tide waves onto the beach. The female first wiggles her body into the sand up to her pelvic fins, where she deposits 1,000 to 3,000 eggs, and then the male surrounds the female and deposits his milk. And it has been observed that up to 12 males have fertilized a clutch. The adults then catch the next wave back out into the ocean, leaving the eggs to develop in the moist sand above the tidal line until they are ready to hatch out with the next tide in about 10 to 14 days that comes to wash them back into the ocean. However, in California, the tidal cycle is mixed semi-diurnal, so if the next highest high tide does not reach those eggs incubating, they can actually have delayed incubation and wait for then the second tidal cycle to come and wash them into the ocean. The California Grunion aren't a commercial species, but they have a high recreational value. People come to the beaches by the thousands to try to grab some by hand. They're allowed to catch in buckets except for April and May. But mainly people come by the thousands to the beach just to watch these large spawning events, and they're a great educational opportunity. Historically, the range for the California Grunion have been from Point Conception down into Baja California. Point Conception is where it says PIC on the map on the left. However, some populations recently have expanded north into Monterey Bay, San Francisco Bay, and Tomales Bay, represented by MB, SF, and TB. They're at the 38 degree north marker. It is hypothesized that the fish populated Northern California during an ENSO event in 1998 to 1999, looking for thermal refugia. If you look at the map on the right, the waters off of LA and Orange County are between 18 and 21 degrees Celsius, but then as you move up the coast, the waters are much cooler until San Francisco Bay that has much warmer waters. So what's the reasoning for having these spawning events on land? Well, there's a lot of pros for this adaptation. You have decreased aquatic predators, increased temperature, increased oxygen, and synchronized hatching that can help with larval success. Although there are some cons to this, there can be desiccation of the eggs, smothering, and there are then increased terrestrial predators. So the egg development has been thoroughly studied by Martin et al. In 2009. It's very easy to study the development of Grunion, California Grunion in their eggs because the chorion, or that outer membrane, is very clear and you can see right through it. So Martin and colleagues found that there were 30 distinct stages of egg development, and the eggs of Southern California Grunion are a little bit larger than those from San Francisco Bay, and those in Southern California also develop faster, and they are ready to hatch at 8 days, whereas in San Francisco they aren't ready for 10 days. And this may be due to the colder temperature and lower wave dynamics in San Francisco. Temperature of the water can have effects on egg development and hatching success rate. You can see on this graph on the right, on the x-axis is time in days going from 0 to 60 days, and then on the y-axis is percent of hatching and survival rate percentage. And at the top you have 15 degrees Celsius, then 20 degrees Celsius, and then 25 degrees Celsius. So eggs at 15 degrees Celsius hatch the slowest, and we're not ready for the first tidal sweep, which could be very detrimental because the eggs are very heavy. So if they're washed into the ocean, they would just sink to the sea bottom and be smothered by sediment. Eggs at 25 degrees Celsius hatch first, but their success dropped rapidly to under 50% at day 28. Day 20 degrees Celsius is the optimal temperature and reflects environmental conditions. Matsumoto and Martin in 2008 measured the effects of salinity on fertilization and egg development. So they found that hyper and regular saline environments had successful gamete fertilization, but in hyposaline environments, only 2/3 of gametes fertilized. Then, after eggs were fertilized in normal 32 ppt salinity, they were then subjected to hypersaline and hyposaline conditions while developing, and only hypersaline environments impacted egg development with a highly decreased hatching success rate, whereas you can see in this chart, hyposaline conditions had the same hatching success percentage as normal salinity, which is likely due to an adaptation for rain events while developing in the sand. So during this delayed incubation or arrested development, these little busters from days 1 through 8 have high increasing rates in oxygen metabolism. As you can see in the chart in the graph on the left is that steep slope days 1 through 8, but then the mean metabolic rate levels out from day 8 forward, and as you may remember, day 8 is the day that the eggs are ready to hatch. Also, oil droplet depletion occurred at a linear rate and was completely depleted by day 39. Also, as the fish developed in the egg, we saw an increase growth in the sagittal otolith and the number of teeth. Also, there was found to be a significant difference in length of a fish depending on if it had already hatched or not. So on the x-axis you see days post-fertilization. Those with open shapes are the fish still in the embryo, and those with a closed shape are hatched out, and you can see that they are significantly longer, but those lengths even out after about 3 weeks. So what is the trigger for hatching? For most other organisms that lay their amniotic eggs on land, hypoxia is the cue due to waiting for a rain event or a flooding. However, this is not the trigger. For Grunion fish, agitation is their environmental cue because that's when the waves would come and agitate the eggs and they can pop out. There were tests done on hypoxic versus hyperpoxic conditions, and actually hypoxic conditions led to lower hatch success because they're in a hyperpoxic situation with a lot of wave action. The hatch event itself can be seen by first an outpocketing of the chorion, so that's just a small dent of the outer membrane, and then fluid begins to leak out of there, and then the fish itself does a twist of its body and a large flick of its tail or tail lashing, and that allows it to break out of its egg, mostly tail first, and this emergence happens within 2 seconds. Sometimes the head is still stuck in the corian, and unfortunately, if it does not make it out, then the fish dies. Now that our little grunion larvae have hatched, what leads to their success? Increased temperature increases their body length, their weight, and their rate of weight increase. Their ideal temperature is 21.6 degrees Celsius, which mirrors again environmental conditions. So the fish does not use its oil droplet until it has completely depleted its yolk. This is due to the oil droplet being necessary in keeping the head very buoyant. The fish, when it is hatched out, it is highly developed compared to other fish, so that it is immediately ready to swim and feed. And also, fish after hatching, they did not enter water cooler than 20 degrees Celsius unless they had completely depleted their yolk and wanted to slow their metabolism. I've already mentioned the impacts of arrested development on fish development and hatch success, but I wanted to talk about its impacts once the fish has hatched out of its eggs. Eggs with delayed incubation of 30 to 35 days had a survival rate that dropped below 80% after just 10 days, and they did not survive past 4 weeks. This 30 to 35 days is past two potential events for them to be swept out into the ocean by the tides. And so this probably reflects an environmental condition that if they have not hatched at this point, that they would not be able to survive out in the ocean. So what's left to study about this fish that has been studied for over 100 years? Well, because of its unique structure of having that very clear corian, and it's easy to raise the eggs in a lab, it could become a model fish for teleost development in early life stages and testing their adaptations. There can also be nutrient analysis done on the yolk content for lipids and fatty acids. Potentially, they have a unique signature. Um, there should be studies on temperature effects on the eggs throughout the spawning season. The data that I showed you about temperature effects on egg hatch success rate was only eggs collected in June. Also, there have been some studies suggesting that coupled temperature and salinity have impacts on hatch success rate. Also, there was a paper written about power plant effluent having an impact on hatching success rate that should be developed further, as there are multiple power plants along the California coast. And finally, when the fish is trying to break out of its egg and out of the corian, it is likely releasing an enzyme called corionase to help break down the corian and have it break out of its egg. However, there haven't been any studies of how that enzyme works on the California Grunion fish. Thank you for watching my presentation, and if y'all have any questions, you can ask me on Wednesday